last panel of the last session of this amazing meeting, and I would like to thank all the panelists and all of the audience for coming and listening and hopefully asking us questions. So I would like to start with asking everyone to just give like 1 or 2 minutes of who you are, what's your background, and what is your take. I don't know how many people have actually read the program, but there are some questions for this session in the program. And basically the questions are, how are we going to seed life and should we seed life? So if you could tell us who you are and what's your take on those questions so we can get started. We can.
I'm Tonya Harrison. I'm the Director of Research for Arizona State University's Space Technology and Science Initiative. So in that role, I do a lot of work with commercial space companies on technology development and figuring out how science and the commercial sector can work together. But my bread and butter is I'm a Martian geologist. So I've worked in Mars geology and mission operations for the last 10 years or so. I worked in operations for Opportunity, Curiosity, and the Mars Reconnaissance Orbiter. And I tend to think about life in the universe, specifically in terms of Mars. Obviously, it's a huge bias, and what we need to think about before we send humans. Our big question for Mars is obviously, is there or was there ever life? And my question is, how sure do we have to be about the answer to that before we send humans? Because regardless of what we do, as soon as humans set foot on Mars, we're going to contaminate it in some way. Even if we're in spacesuits, we touch our spacesuits to put them on. We're going to be touching things that are interacting with the atmosphere. There's going to be some exchange with our things that are in the HAB with the atmosphere. So there will be contamination the minute that humans set foot on the surface. And so how sure do we need to be that there is no life on Mars before we say, okay, it's okay for us to go and do this? Or what do we need to do to be sure that there maybe was life on Mars, and how do we protect ourselves and life past or present on Mars from that?
So should we contaminate Mars?
I think yes, from a geologist standpoint, because I don't think we're going to be able to answer all of the key questions we have about Mars without sending humans. There's only so much that robots can do. The rovers are wonderful, but they're also incredibly limited compared to the human brain.
Hey, I'm R.P. Oates. I'm a program manager now at Rhodium Scientific. We deal a lot with taking biological things to the ISS and back for now, but hopefully it should be extended out to Orion and even further later on. But I've only been there about 3.5 months. Previous to that, I did my postdoc in the Department of Defense, specifically at the Defense Threat Reduction Agency. If you're not really familiar with their mission, we do a lot of counter weapons of mass destruction development, especially with detection and nuclear monitoring and medical countermeasures for those that operate in nuclear environments. And one of those things, I'm a toxicologist and chemist by trade, but I masquerade occasionally as a radiobiologist, and I'm very fascinated with ways in which certain organisms can respond to radiation, and especially different types. I think we've talked a lot at this Discuss conference at the Breakthrough, really about what are the chemical requirements for life. You know, there's a lot of biochemical, biological requirements for what we would call life. One I think that's really less thought about is what are the electromagnetic requirements for life. There's a lot of papers that have come out in the past 5 or 6 years that would suggest that there's an electromagnetic component or an abiotic component to life. One particularly cool example came out in 2014. They had some stem cells that they were able to introduce low-frequency electromagnetic fields to, and it changed the way it was able to differentiate. So what that could possibly mean, if we're to immigrate, what does that mean for SETI? If we're to immigrate life on Earth, are there different electromagnetic requirements depending on where we go? Are they gravitationally dependent? Or are they mixed field radiation dependent? You know, on Earth, we're pretty much used to a certain electromagnetic milieu. But up in space, if we're to take stuff off planet, like we're talking about doing, there's a mixture of chronic low-dose radiation. How do we get through that? If we're going to be out there for quite a long time, we have to be able to understand what are those responses and how do we overcome them? And also, I think a lot about what that can mean for us. Can we use some of those responses to make ourselves a little more radio resistant if we do indeed migrate ourselves after that? So that's my background and where I approach immigration.
I'm Megan Palmer. I'm a senior research scholar working at the Center for International Security and Cooperation, or CSAC, at Stanford University. I'm a bioengineer by training, but have spent the last decade working on policy, research, and strategy for a number of different organizations, mostly focused in synthetic biology. And most of my work now focuses on the understanding the ways in which individuals and organizations contend with the impacts of their work, especially in and around emerging technologies and advanced biotechnology. And so the biggest sort of experimental test bed that I work in right now is an international competition in genetic engineering that has been going on for the last 15 years. This year we'll have probably about 6,000 students in over 300 teams across 4 dozen countries that will be developing genetically engineered machines for useful purposes. And so I both direct the responsibility part of that competition and also study the ways in which these individual scientists and organizations understand what is a useful purpose, and also how we adapt our policies over time as we develop new practices and new ambitions. And so while I'm not an expert in space ethics and space policy, I think my contribution here is a few different topics, so 3 in particular. As what I understand is, I think there's a revisiting of what is sort of planetary protection principles, policies, processes, as we think about new types of ambitions, new types of entrance into this space. And I see them really mimicking a lot of the types of puzzles that we see in other types of biotechnology here on Earth, especially areas like the development of gene drives that could potentially wipe out species, or technologies that could bring species back to life. Secondly, I think there's some really interesting puzzles around private actors and investments that call upon us not to sort of back down, but actually to really understand how to safeguard the enterprise as a whole. And third would be around a research program that we actually developed here along with several other universities around biotechnology, in thinking about the future policy landscape there, where we had to think about the paradigms we were using for safety and security that were all based on containment. And we had a research program focused on how can we think beyond containment in a world in which we may never have been contained in the first place, and where our ambitions may actually extend beyond a contained environment. And I think that opened up a lot of opportunities to think about the tools to monitor and actually understand intentions of containment and what we may have already done through our actions in the past. Thank you.
I'm Andrew Pohorille, and I'm by education a theoretical physicist. I have been at NASA Ames for over 20 years, and on a faculty at UCSF even longer. And according to Dan Golden, who was the administrator when astrobiology started, I am the original theoretical astrobiologist, and he says that because when he requested that Ames develops a program in computational astrobiology, I was given this task, and I had no clue what it was supposed to be, and quite frankly, so I kind of was a failure, and quite frankly, I still am not sure what it is. One thing that I know for sure is that it's not a very good idea to separate theoretical and computational studies from experiments, and my main interest is at NASA is in studying the origin of life, and I'm very fortunate to work with a group of very capable experimentalists on this, and I think that that's the right thing to do. Otherwise, I develop some hardware for spacecraft hardware for biology. I am the co-lead of the Center for Life Detection at Ames, and but also I have been for over a decade. I was trying to advocate this program that is an alternative to seeking a separate genesis of life, which is to take life, microbial life, to space, our microbial life to space, and try to adapt it to different space environments. And that's kind of similar to what you are interested in, but I would advocate a more balanced approach, which involves both evolution and re-engineering or engineering, and both have their strong and weak sides. Evolution has this strong side that you don't have to know how systems work. Evolution will take care of it. But the problem with evolution is that you may not have a viable path, evolutionary path from the point that you started, let's say terrestrial life, to the end point at which you want to find yourself. It's a rugged landscape, and there are those theories that say it's kind of like a storied spherical horse theories that say that a full optimization should be possible, but every experimental proof says something else. On the other hand, re-engineering or engineering requires a lot of knowledge and anything, and any weak link is going to break your system. So it's a very ambitious undertaking, and I think that both should be somehow combined when we try to take our life somewhere else. I was not very much thinking about what we can do, what practical things they can do. I was more thinking in conceptual terms. If we can adapt our life to a number of different conditions, then it probably something like that exists in space. And but I have a full folder of white papers on this subject, but I haven't made very much headways, which tells you that I am a very poor salesman, so it's good that I didn't choose that as a career. Thanks.
Hi, at the risk of sounding like an AA meeting, my name's Lynn Rothschild. I've been at NASA for over 20 years. And what are you supposed to then say? I don't know, at these AA meetings. And I'm also faculty at Brown University in molecular and cell biology. I'm a biologist. By love, I work on protozoa and algae. By early training, evolutionary biology. By my later training, adding on molecular biology. And then when getting to NASA, becoming heavily involved in starting and then being heavily part of the astrobiology program, which is really a fantastic experience for an evolutionary biologist because you're no longer confined to one planet. You can start to look at life as a universal phenomenon. And so I particularly got involved in life in extreme environments, not because I had this great desire to, but because I was looking to reenact what a day in the life of the Precambrian might be like, and so you end up going to extreme environments to get rid of, as one of my advisors used to call them, big people organisms. I don't believe in saying higher or lower. And so next thing you know, you're mushing around in Yellowstone and the Rift Valley in Kenya and in New Zealand and in Australia and the radioactive springs and all sorts of wonderful places like Bolivia. And so I was perfectly happy looking for life in the universe and mucking around. And who said the other day, they always wanted a job where you can wear blue jeans, and that's been my goal in life. And then about, I guess it was 10 or so years ago, I'm looking for him, Pete Worden asked me to start a program in synthetic biology for NASA, believing, and I think he was quite right, that synthetic biology is the enabling technology there is to bring life off planet. And I've actually come to believe that, Pete. I think you're absolutely right. And so I sort of dusted off my background in molecular and cell biology, and we've been going back and forth to Washington and the world. Oh, are you choked up, Andrew? This is very touching. We work on the same floor. I thought he'd heard the story before. Anyway, so we've sold the program back and forth, and I'm now on my second tour of duty representing NASA on a synthetic biology White House working group. And we've gotten involved in developing various biological technologies, including using fungal mycotecture to build structures and biomining organisms to pull out metals and so on, and to bioprint and so on. Many of these projects, thanks to Ron and others in the NIAC program, have been funded by the NIAC program at NASA headquarters. So those are my various things, and also little known is one of the early incarnations of the NAI. I was leading a project, oddly enough, on panspermia.
Thanks, Lynn.
Let me just add one last thing, because I should have remembered this, is that I am also a PI on a satellite mission, which is currently active with the DLR, where we have organisms right now orbiting the Earth, and it's a very unusual mission. It's a Eucropus mission in that the satellite started off not spinning, so we were able to do a set of experiments with microbes doing basic things like transformation and looking at promoter strength and so on under different gravity regimes. So we started off at microgravity, and that occurred, unfortunately, during the furlough. And then we've in the process of mimicking lunar gravity right now, so the spacecraft is spun up, but because we had the spacecraft balanced, we'll get another shot at lunar gravity in a couple of months, and then a shot at Martian gravity. So we have those data coming down right now.
And you should also say you're actually trying to send life to Mars with your iGEM kit.
Oh yeah, well, Megan mentioned the student synthetic biology competition. I don't know if you mentioned that it's called iGEM, but since 2011, I've been the faculty advisor of a combined team of primarily students from Brown and Stanford universities. 1 year we had Spelman involved, we had RISD last year, and this year we've had some students begging from Princeton. So we're now, since we had to get it into 20 letters, we're now Brown, Stanford, Princeton, somehow taking out the last few valves. And we've got another, I'd like to think, exciting SynBio for Space project going. Kate's actually helping our team on that.
So to start, I want to just get that question out of the way, probably the most obvious one that we haven't actually answered. If we decide to actually want to send life somewhere else, what are we going to send, and why not a bacteria?
Well, I mean, you sort of have to first ask the question, do you want to send life, and why? What's the purpose of it? Because even though I just said that I'm very bullish on using biology as the enabling technology for human settlement, in my astrobiology outlook on life, there would be nothing more tragic than if there was life elsewhere and we were not able to detect that because we've contaminated the planet with our own life. And so I'd like to think that the science takes priority. If you can do both, that's even better.
So that kind of goes back to what Tanya was saying, that if we ever land on Mars, then we basically contaminated it already.
Right, so trying to think about, do we want to take that risk before we definitively answer that question or not? But then you also have the question, can we definitively answer that without sending humans there? And I think of it from the standpoint of, if life ever arose on Mars, it probably did so when Mars was warm and wet and a much happier place for things to survive. But that was also about 4 billion years ago. And a lot of stuff has happened geologically on Mars since then. You've had volcanic eruptions, you've had tons of impact events, you've had wind, maybe you've had rain. And so any of that evidence of life is probably very deeply buried. And so I don't think we're going to be able to get to it with rovers that can drill a couple of centimeters into the surface. And I don't want to kill the Mars program by saying that, but I don't think the missions that we have are going to find any evidence of past or present life because of that.
So basically what you're saying is we have Schrodinger's life on Mars.
Exactly. And I think in order to answer the question definitively, we're going to have to send humans, and we're going to have to drill giant cores akin to ice cores that we have in Greenland to really see what the geologic history of Mars has been.
So should we do it?
I think you made a pretty interesting and compelling case in your presentation of developing tools to really systematically understand the ways in which we are contaminating our environments as we enter into new spaces. And what we've seen here on Earth with new genetic technologies is thinking about technologies that might allow us to do better forensics over time, do better detection, do better cleaning, that could be coupled to this. And so I think we shouldn't necessarily think about it as an either or, but how do we develop the sophisticated technologies, policies, capacities, and processes really at the end of the day to be able to really understand in lockstep what exactly we're doing when we go there.
But if we go with, if we take life out beyond Earth, one of the reasons is to learn something very fundamental about life. I mean, we can focus on growing potatoes, but that may not be necessarily the most exciting thing that I could think about.
You can distill them.
Yes, that's very useful. I mean,
Ooh.
Wow.
We both come from Poland, so it's a natural association. So don't be surprised about that. Life taken away from the Earth is exposed to stressors that are very different than those that are on Earth. We really don't know what happens to a biological system that finds itself in this kind of conditions that it has never experienced. And so we can learn really some very basic things about mechanisms of evolution. But the sad part of this story is that we essentially have great tools to do wonderful things with microbial biology and genetics on Earth. But we have almost nothing to do it in space. And there is really no will to develop these kind of tools, even though it's possible. And until that happens, this is really, everything that we're talking about here, you talking and so on, it's just a dream.
Well, I think there is a lot of will within the Department of Defense to make biotechnological tools in resource-limited environments. So that does happen, and it's happening now. I'm not sure that that's well advertised as as it usually is not. But some of those same technologies, I think we could at least go through the thought experiments of risk and governance, like you were mentioning too, Megan. I think it's an important point because commercial space travel is happening. We're going to go up there. There's going to be tourism there, and that's going to happen. So whether we like it or not, life is going out of the planet. It may not go very far, but it certainly is. So what would happen, for instance, there are experiments that are ongoing now that do kind of test for, is something on your skin, is it more or less pathogenic if it's introduced to microgravity or mixed field radiation in a chronic low-dose way for, say, a certain amount of time. I think that would be very interesting. So what about if it got back to Earth? Would it be seen as, is this bug going to be a pandemic? Is it start some type of transmissible disease? Can it happen? I don't know. So it would take kind of proving that out, while we still can, much before we continue to emigrate off planet. I think it would be wise to put that through the paces as a community. I think that would be very interesting to do too.
I think that's an interesting point. Maybe the Andromeda strain is something that we impose upon ourselves, and we haven't really thought about that.
Do you think there is, in general, a lack of really conceptualizing, what both the awe at stake here, as well as the sort of practical realities of things that will eventually occur as this, as the ambitions in this space and the diversity of actors increase, right? We talk a lot about, the difficulty of making technologies, of doing the science, but there is a really difficult process of individual and collective imagination and planning around what we do with the information that we collect, right? Even our conversations so far, right, that conception of what if this contamination has already occurred? What if extraterrestrial life is already with us? Is something that I think is not a commonly held conception, right? And so we have an obligation to introduce that to others, to wrestle with it, and think about how we manage this type of governance. And it certainly isn't easy, and others here have said that it may be harder than the science and technology itself, but it is certainly worthwhile, and if not the most worthwhile thing to do.
But I would say that it would depend on very much what this life is. So say you found that there was an alien life on Earth, some sort of bacterial species or whatever. You might say, ooh, get rid of it or cage it or shoot it and stuff it or, something like that. But what if I, discovered in the lab that your mitochondria were alien? What are you going to do about that? Not much. You would have to be able to live within your own skin. So I just throw that as a thought.
So is it still alien if it's been
That's
here?
right. And that's really sort of my point. If an alien life form has been here for a long period of time, it's already been incorporated.
It's not alien anymore.
It's no longer alien.
I'm not entirely clear what was the meaning of the word alien. I mean, we have pathogens that are extremely dangerous, that are not alien, but they could cause enormous damage. And if there is something alien and doesn't do anything bad, I mean, what's the problem? And what does it mean it's alien? So it has DNA that you cannot PCR, or what?
Yep.
What was that?
So in that case, you can think about it in a way that if it's capable of interacting with our biosphere, that means it can eat the correct chirality amino acids.
Yes.
It can read our DNA, or it can have any kind of nucleic acid that our cells will amplify, then it's not alien.
But are we going to speculate completely? I mean, is there a shred of evidence that for this kind of shadow biosphere? I mean, is it anything beyond the fact that Paul Davies said so?
Thank you, Anne. Let me also clarify your point, Kate, and that is, to an evolutionary biologist, there's a total difference between convergence and common ancestry. So you could come up with an organism that has exactly the same DNA, exactly the same proteins, exactly the same lipids, exactly the same operating system that you made in the lab, or that was originated somewhere else and came to Earth, that was indistinguishable. But would we consider that the same? Well, it would not be on the same phylogenetic tree because it was not part of the same continuum. And that's something that then we would have to grapple with as evolutionary biologists. It would be us, but it wouldn't be us, if you know what I mean.
At that level, can you tell the difference, though? As a non-biologist, I don't really answer that question.
That's a tough question, and then you'd have to start clinging, and that's what I think one of the issues synthetic biology brings up. You have to question whether you need to cling to the notion of having an unbroken physical instantiation going back 3.8, 4.2, whatever billion years, or whether you can recreate a mimic, and that's good enough.
Well, that depends on your goals. Obviously, if you're interested in history, if you want to get this phylogenetic history of life in an environment, then it does matter. But if you want to grow potatoes and then transform it into vodka, then it doesn't matter.
You do that, go straight to the vodka.
Well.
So would it matter to you, Megan? Like, let's say there is a life that came from somewhere else, but it's so similar to the native life. Let's say we design a synthetic cell that happens to be just exactly like the inhabitants of Europa, and we dumped it under the ice on Europa. Can I do it, or can I not, or should I not?
Probably not.
I think it highlights this interplay of, when do you know, right? How do you know and act at the same time? That's fundamental to all of these conundrums, right? And that there has to be some sort of sequencing amongst it, and that the world isn't static and may have acted before you get there in the first place. Sort of returning back to whether it matters on knowing, if we are alien or part alien. I think that fundamentally, yes. I mean, many of the biologists that got into this area are interested in this area because it's not just beautiful, but it questions fundamentally who we are, our understanding of our place, right, in the universe. And so, yes, I would say at least for a large portion of people that matters quite a bit, and it's a value in its own. It's less of an outcome oriented than anything.
Did you feel? Would you feel differently about yourself if you found out that you are a Martian?
Yeah.
I definitely would.
Yeah.
Well, how many people felt differently when we started finding out that probably everyone in the room has some Neanderthal DNA? I mean, I don't know about you.
And I mean, at some point, it doesn't really matter because the alien will start interacting with the environment and changing the environment too. So how do you tell it's alien anymore? Like, I came to this country and I brought my favorite pierogi, my favorite food. Now I'm American, but pierogi are still my favorite food. Yeah. So if life comes to another planet and brings certain kind of metabolism that didn't exist on that planet before, is that how long until that kind of metabolism won't be alien anymore? That's just what the planet or the moon does now. And can we engineer it somehow?
Well, I think just another comment on here. I think these questions, really sort of existential questions on what is, our being, are also paralleled right now in a lot of conversations on what we should be preserving in terms of our diversity here on Earth, right? Our biological diversity here on Earth, and how we understand our connection, as you said, to Neanderthals, but also things that are around us today. So as we're thinking about how we collectively make decisions on what to preserve, it's already here. We can find the seeds of potentially productive and non-productive discussions around those topics.
Well, I kind of want to posit something that is perhaps contrary to what you were saying and contrary to some kind of undercurrent of this meeting, which is that I think that we know relatively little about how cells really work. And we kind of understand perhaps different parts. And I remember that a number of years ago, we wrote this paper with Dave Deemer when we were kind of saying, individual parts we kind of know how they work, but how they work in concert, we don't. And there has been obviously progress since then, but we still don't understand most of it. And we can look at this exploration of space as an opportunity to learn more about these whole processes, and in both operational sense, but also in an evolutionary sense.
I mean, I definitely agree we don't know much about natural life, but that, yeah.
No, saying Megan just sort of touched on the idea of preserving the species we had, and so I'm just going to use that as an excuse to say that about 2 months ago, I was in an Intelligence Squared debate in New York on the question of de-extinction. And people brought up the fact, oh, it doesn't matter if the last 4 rhinos go extinct because you just bring them back. And my reaction was, if you can't preserve the last 4, what makes you think you can go through all the trouble of bringing something back and then preserving it? And it's sort of like these arguments, and I believe I said that in public, that people say, well, Mars is plan B. We just go there if we screw up planet Earth. Well, I think there's been a little bit of dancing around that, I feel, in this meeting. And the fact is that the worst place you can imagine on planet Earth is still infinitely better than plunking yourself down on Mars. Mm-hmm. That this is not just plan B.
Mm-hmm. Yeah, Lynn, I think
you're talking about, I could find you organisms that could live on Mars or Enceladus or Europa or whatever, but, you're talking about sort of dealing with life as in a one-dimensional thought process rather than what does it really take to live there?
But do we really want to go to Mars because we're preparing this as a future real estate for us, or is it because we just want to know? I mean, just for
I want to know, but there are people who feel this is, they say, this is their future real estate.
Yeah, it's just going to depend on who you ask.
Yeah.
Yeah, I was just considering that concept of rewilding, where you use genetic material to reintroduce that into native populations, and our track record for doing so is arguably pretty bad. So if we can't do that here, what makes us think in a practical sense that we'll be able to do that exoplanetary? But it is an interesting test bed to see if we could do it. That's why I find the work with synthetic cells to be so interesting, just because there is zero reproducibility. There's really no predictive context to it. So if we could build that here, I think that would be a nice way forward to at least have some type of predictive capability in biological systems that we can rely on and in different environments. I think that would be a great thing indeed. So anything I can do to help, I'll
really
try
good
to.
point. Some of you may know the artist Daisy Ginsburg, who's based in London, actually has a project right now going on called The Wilding of Mars. And she's talked to various people in the planetary science, biology community, and so on. It's a very cool project. Right at this point, it's very conceptual, lots of drawings and so on, but it would be nice to move it into an experimental phase.
I just building off of your comments on the synthetic cell, I think it's an example of an area where there can be a lot of innovation in breaking down the steps that we would want to know in terms of, increasing our understanding both of, fundamentals of life here, but also, not having that same type of contamination imagined, or being able to do continuous monitoring and experimentation somewhere else that mimics some of the types of regulatory innovations and technologies that are coupled to it here, again, on Earth that we're considering, including in places like gene drives, for those of you who may not know, that allow for the propagation of traits across populations at more than normal Mendelian inheritance. There's been a lot of thinking about, how do we actually do this type of experimentation? How do we learn from a technology standpoint and from a policy standpoint in developing up these new types of experimental testbeds and technologies that are sort of semi-contained or have stages in their experimentation? And so I think that's a really great way to think about, how can we begin to be more predictive? How can we leave different types of traces that can be uniquely detected and differentiated from the things we might accidentally leave? Allows us a way to, a path forward to not always say we're always going to be bad at rewilding, but what would it actually take to get better?
I think during this whole conversation, a couple of people brought up interesting points where you didn't use the word leveraging, but that I think that a lot of the things we're talking about are very unlike most of the other sessions the last 2 days, particularly the first ones yesterday, in that the primary driver for these fields are not space exploration, NASA, and so on, but are other sorts of terrestrial-based applications. Megan is, I'm going to go out on a limb here and say, the world's leader in looking at the ethics of synthetic biology. And you've got the experience in the Defense Department, and Kate certainly does the synthetic cells and so on. These are the drivers, the medical community, agriculture, and so on. And so we're very fortunate in these areas that we're able to leverage off these other discoveries and move them into space.
Yeah, I think that's a very important point, Lynn. I think it's just now as a community that we're all really starting to understand all the dual use and dual purposes of what these types of technologies can bring to bear, especially for society here on Earth, and what we can do as we move forward in space travel. One thing I was just kind of thinking about as we were talking is maybe from a bit of a societal standpoint about what we would be comfortable with to as we immigrate. Because if would I eat something that contained a gene of its own species? What about if I had to insert a gene of a different species into that, and then do I still eat it? Sort of the concepts we think that surround GMOs here on Earth, but what about in its extreme? Would I eat something that's completely synthetic? Would I do it? If I was traveling all the way to Mars? I mean, it seems like a really basic question, but and it kind of is. But to be honest, would you be comfortable with that? Could we do it? And how would that methodology, would that translate to us being here as a terrestrial species? Just like you'd have people out there trying, pioneering things, and would they adopt it or would they not? I think that's an open question too, something to consider.
I mean, there is really no doubt that in terms of these dual issues, that we can, for space applications, we can learn a lot from what's going on in terrestrial biology. I'm less sure how much people who do terrestrial biology believe that they can learn something from doing experiments in space. Probably they don't believe that they can. What makes it even worse is that Pete was saying that he was almost fired for for trying to start this synthetic biology program, and suddenly he was, happened not because everybody thought that it was such a great idea. You don't fire people for that. I am not entirely sure how space agencies, especially NASA, sees that. Is it really a value, something of value, or more of a problem? There is this planetary protection issues, and what are we going to learn from these experiments? Are we going to take this bacteria or whatever microorganisms to space, try to look at them? It costs money. Will we get a better mousetrap from that? I remember that Pete asked me once to prepare some kind of a report for him. Can we use microorganisms to extract fuel, oxygen or hydrogen from regolith on the moon. So if we could say, yes, you can do it, then NASA perhaps would see value, although even then I doubt, because bacteria are kind of unpredictable. They can die, they can mutate. If you have a physical chemical system, that's something that is predictable. So I'm not entirely sure whether there is something in the cards that will really, some kind of practical political motivation that would really let this idea of migrating our life beyond Earth to develop into any kind of coherent program.
I think it's very touchy politically because you're going to get a lot of people questioning whether it's playing God, and so I bet a lot of governments just don't want to touch it at all. Maybe that's a question for the policy person.
So at some point, I see the cubes came out. So at some point, we might want to include the cubes in our discussion and the feedback and the fuel from the cubes.
Not to be confused with cube.
Yes.
So I suggest we deploy the cubes.
Okay.
Okay.
Cube set.
So we go from side to side, cube to cube.
Suborbital.
Bonus points if you incorporate a toss. Yeah.
So are you speaking to this? Okay, so this is going to sound more critical than it's intended, but the topic of this session is emigration. And I think because we're mostly scientists here, it's mostly been about exploration and respect and don't contaminate. Emigration is really driven as there are a number of immigrants among us because you don't have something where you are that you want, and you think you might get it somewhere else. And it's not primarily about discovery. It's not primarily about research. And in fact, historically on the Earth, emigration has resulted in catastrophic contamination. They're totally intertwined. So I think real emigration will start when the possibility of regulation against contamination is eliminated completely. And suggestions like using badminton shuttlecocks to deliver promiscuous spread of bacteria will be done without any sense even of what you might be contaminating. So they're very different topics, it seems to me. And I think we're struggling right now with Mars, largely, and possibly a couple of other bodies in the solar system. And that's appropriate. But I think I agree with whoever said it. We're not going to move, you said it, we're not going to move to Mars, right, in any significant numbers, because it's so different. So I'm not quite sure where emigration really enters into this discussion.
Well, sort of, the counterexample, not worrying about the planetary protection, as I think was mentioned earlier, is the moon. You can, the planetary protection people say you can just go, have fun. And if I can just add a quick comment. People talk about it, then if we do immigrate and then we have these societies built and it's going to replicate the Earth and stuff. It's like, no, the people who go ahead and emigrate, whether they're coming to the New World or they came out of Africa or they go off planet, are the really adventurers, the risk takers, sorry, the second-borns, nothing to lose, and they don't care if they die off planet. And so you're going to get a very different, at least first few waves, I think, of people there, and a very different way of thinking than someone like me who is much more risk-averse, probably, than that.
I think, Charles, there's another reason.
So I had a couple of different comments. The first one, there was a question about whether or not people think that there's anything to learn from going into space about life sciences here. So the experiment that I performed on the shuttle was called frog embryology, and it was out of Ames. And it was looking at the organization of embryos to see whether or not there was how gravity affects organization. And we found that it does affect organization of sort of cells. So there is some basic biological research that definitely can benefit from microgravity. But the question I wanted to ask has to do with the comments about synthetic life and containment, and some of what seems like we're assuming that we're only concerned about containment of possible bad effects on other planets. The question, without sounding like an alarmist, is do you have any concerns about the idea of synthetic life and having it done widespread here on Earth in terms of how it would impact life here, After all, we've restricted smallpox to just a couple of locations. The more we do and the more that we learn about organizational structures, how life, the robustness of life, the robustness of organelles or packets of whatever you call, how do you look at that in terms of containment here and going forward?
I must say I find that question extremely flattering because that's assuming our fake synthetic cells are actually good enough to do something bad. And
well, it's not necessarily now. I'm talking about going forward because we have these concerns, and I'm someone who would be, what did you call it? I'm the third child, so I'll do anything, right? And have worked with around Lassa fever and some very dangerous diseases in West Africa and other places, and understand and feel very comfortable. But I feel like there's a sort of a glibness in some sense about the possibility of impacting life here, not within the next 5 to 10 years. I know that we're very far from that. But when we talk about inserting other kinds of proteins, we continue on and talk about these things. I think we have to look at that as well. And perhaps space platforms are a wonderful place to do some of this research, because you do have the ability, once we get that far along, to be able to test things without the potential for creating something that might outcompete some of our very simple microbes.
I can speak to this a little bit. One of the reasons that we've become so interested in this genetic engineering competition as a test bed is to understand and develop a place in which we can monitor and develop adaptive policies on the types of risks that might emerge from the new ways in which we are doing science and engineering. And in particular, really beyond those categories of lists of concerns and particular species of concerns or specific experiments of concerns, and really have a grounded way to look at over time what are the things that we might, that might cause harm. And not just a particular type of harm in terms of safety, but other types of harms as well.
And the other reason I bring that up is because when we may be talking about timeframes within our labs of looking at things for 10 years, 15 years, whereas the history of microorganisms is much longer than that, and the adaptability and what happens over a time scale is much longer than that. So just really keeping those things in context.
And in many cases, we've been looking at the really short time scales. How can we get beyond updating policies every 5 or 10 years to updating them every year when we see a lot of advances? But I will completely agree that thinking about long time scales and the repercussions that we may not see for a long time is something we're still grappling with. This is a very live topic. In addition to even how we share the information and the products of our science and engineering. And this is something that certainly deserves just as much attention in innovation and policies and practices around it as innovations in the science and engineering itself. And figuring out how to couple those to a grounded reality of what we are doing is part of not just the challenge, but really the opportunity here.
Just to be clear, when we finish an experiment, we don't go outside with a test tube and just dump it in the driveway either. Obviously, you're supposed to have it contained. And I know at one point DARPA had a program, or we're talking about a program to have people engineer kill switches, things like that, that there are ways to build in safeguards. I know Megan's been heavily involved in that as well.
Yeah, I'll just mention one of them. So I think DARPA's Safe Genes program is a particularly interesting example where they've been trying to develop ways to monitor, to develop ways to switch on and off the types of effects that we might want to see, ways to actually reverse those changes over time, and treating that as the central design challenge integrated from the start, rather than something that is on the side, or you develop the technology and then the safeguard afterwards. And I think we're going to have to see more and more of that. And there are also teams of folks who are working in the social sciences and policy who are integrated with the scientists and engineers on these teams. So we're going to need to see more and more of that.
So we have a next cube, I believe.
So speaking about timescales, let's go to the billion-year lifetime of the universe timescale. Isn't there another reason for emigrating, which has to do with survivability? Maybe the same reason that you plant marigolds in your garden, or us as a species rooting for our ecology would like to be able to guarantee that a benign evolution somewhere else in the galaxy doesn't crowd us out. And so if you go to that long time scale, I think it's also relevant to the Fermi paradox, we have a really good reason to emigrate. And that is that if it's about the survival of our ecology, we'd like to see it propagate so that something more dangerous doesn't appear where we couldn't actually have a positive influence.
I hope it's not an experience that anybody in this room will have. Maybe somebody in 500 million years ago from now, but yes, I'm not sure how much I would worry about those who come 500 million years from now.
If we still have humans in 500 million years, we're doing
well,
pretty well.
probably not. Probably not.
Yeah. Well,
there will be some other species.
But I think that does bring it up an issue, again, the idea that, well, we'll just send some people to Mars or we'll send some out on some spaceships. We haven't evolved to be generous. We've been evolved to have self-preservation. Who is going to be the person that decides who gets to be on the lifeboats and who isn't? And so I personally think, and I know this will no doubt engender a large round of laughter, but I think we're going to have to start nudging the Earth out as the sun gets hotter, unless you're going to do something about the sun and turn Earth into its own spaceship.
Well, to the extent that the singularity is 42 years away.
My ears must be going. But I firmly believe that.
Yeah. Okay.
Maybe I'll just make one quick comment. I do think that these questions about the types of catastrophic and existential risks that we might face here are currently shaping activities, investments right now, right? And so it is not just a question of how do we preserve, right, our own diversity and the Earth's biodiversity in going elsewhere, that we're thinking about this, but it's definitely shaping things that are happening today. And so I think it's definitely one of many worthwhile inquiries to really heavily dive into. And what I'm interested in is how do we actually couple that theory, that philosophy, those values in a coupled way into the types of programs and strategies that we want to see. But I think there's many, we live in a global pluralistic society, and we're going to have to contend with that as we're developing, these endeavors.
It is a known societal problem that has been discussed on many platforms many times, which is what do we do with situations that are very high risk but very low probability? Do we prepare for those? I mean, a typical one is okay, what if we know that there is an asteroid heading our way? But there are many others there are medical issue problems of this sort and so on. And so far, that was a worry, but so far there is no policy, or the policies do nothing, essentially.
It's hard to combat optimism bias, and I don't know how we get past that as humans or as a species.
So I see a cube over there.
Yeah, I've got the cube. As we try to understand biology, life as we know it, we've got this really difficult problem of just one example. So trying to figure out what is necessary versus what is contingent is a real conundrum, and I always assumed we'd need a second genesis to help us along. But I'm wondering, with the work that you're doing with the synthetic cells, can that get us anywhere closer to untying that knot?
Exactly. I always say synthetic biology is the art of the possible. Jill, you're absolutely on the nose.
And making impossible possible.
Steve gave this talk yesterday, which was, he was not sure whether I was listening, but I was listening. So, which was essentially, okay, the second genesis will be in the lab. And there is something that we can call universal biology, because laws of physics, principles of chemistry are universal, and that constrains what's possible in biology. So there is something like universal biology, and it doesn't necessarily mean that we have to go somewhere else and find second genesis, third genesis, 20th genesis to determine what this universal biology is. We can, at least in principle, solve this problem in terrestrial laboratories. You agree, Steve?
Yeah, I would wait for the cube to say more. And of course, there may well have been many genesis of life on the Earth. What we're just seeing is the tip of the iceberg right now, what was able to outcompete everything else.
But to the point which was made about the need for survival, I mean, is there not consensus that life on this planet will disappear in something between 100 and 600 million years, and ironically, because of the level of CO2 will decrease? So, I mean, is that not clearly that we will disappear? So, the need to survive may be useful.
If there's no one that wants to answer him. I'd like to answer some remarks that have been made here, because there's a misunderstanding that's being voiced, and we're not going to Mars to escape from a dying Earth. In other words, the purpose of Mars is not to say, well, we have a planet B, let's just let the Earth be destroyed. Okay, that's not the point at all. Okay, the point of settling Mars is to create a new branch, or perhaps dozens of new branches of human civilization, that will add their creative power to human civilization. Okay, you don't have children so you can kill your brothers and sisters. You have children to bring into the world new creative forces. The development of Western civilization in North America, while some Europeans did emigrate here and they did save their lives, but they were only a tiny fraction of the actual European population at any one time came here. But they created a new and dynamic branch of Western civilization that was able to do a noble experiment, putting into action the ideas of 18th century liberalism, and invent steamboats and telegraphs and electric light bulbs and centrally generated electrical power and aeroplanes and nuclear power and the internet and all this stuff. And so it has greatly advanced humanity as a whole to have this additional member of the human family. And the idea of creating human civilization on Mars is not to abandon the Earth. It's to expand the power of humanity by adding new nations. It's together to Mars and then together with Mars. That's what this is about. And the purpose of going into space more generally is not to escape the Earth so that when it gets hit by an asteroid, some people are there to watch. It's that if we become spacefaring, our powers over nature will expand and we'll be able to divert asteroids. That is what the purpose of this. And the purpose of being able to create new species is not so that we can destroy the old ones, but to enrich the world with many new and wonderful species. And
so maybe we, there's
Robert,
too many questions, maybe we can start answering.
You misunderstand me. I agree with you. I'm just saying that there is that feeling out there that I have heard expressed, that that's why you go to Mars. I'm not saying that that's the right reason or the only reason. I agree with you that those are better reasons. I'm just simply expressing that there are people out there who say that's why we go to Mars.
Those people are not confused. And as a person who is very much interested in human settlement of Mars, I say, they're not on our bus. I mean, and then finally, I would say the following, okay? If you give a teenager a car, he will wreck it. If you make him work a part-time job and earn the money to buy the car, he'll take some care of it. If you make him build the car himself from a wreck, it will be absolutely precious to him. Okay, we are callous about taking care of the environment of the Earth because we didn't have to create it. Okay, we never had, it was given to us. And so we have an attitude that is somewhat reckless with respect to it. But if we actually have to undertake the work of creating a new biosphere on Mars, I think this will very usefully transform our views on the value of a living nature. Similarly, if we have to create new species, we understand the value of a new species because we did the work to create it. So I don't think that these enterprises are in any way counter to the goals of preserving the Earth or preserving the natural environment of Earth or preserving species on Earth. I think they're very much in tune with it.
Thank you.
Yeah, as a lifetime fully paid up member of the Mars Society, I have to agree with everything that Robert Zubrin just said. So, but one other thing you didn't mention or didn't repeat, you said it in your talk, and that is that there is an inevitability of migration that goes to the people who are going to migrate. And this is sort of a Darwinian tautology, that if you don't survive, we're not going to talk about you. If you do survive, we will. If you do migrate, we're going to find you. And I'm not sure where we heard that life is going to go extinct in 100 million years or because of carbon dioxide or anything else. I mean, the Eocene thermal maximum was 9 to 14 degrees warmer, depending on where you are on the planet, than it is today, contrary to what you hear in the political press. This is not the hottest year on record, and not even close, and life did just fine in the Eocene. In fact, Professor Prothero wrote a book calling it the Garden of Eden because, of course, there was life in Antarctica. But I want to go back one step. So you got to keep in mind that, yes, we don't, as you are doing with your artificial cells, as we're doing with artificial DNA, as many people around this world in the community that you mentioned are doing, we are learning what is necessary for what is possibly universal, what is possibly contingent in biological structures, all the way from the molecular level to the cell level. I think that's a perfectly good alternative to going out and trying to find a second example. It just is not culturally as interesting to the community, not to my mother, not to the population as a whole. So I don't think it's a real problem in terms of understanding. What will be difficult when you go out into the world looking for odd life is you won't be able to recognize it very well. And I think that's where, so what's wrong with doing synthetic biology in the laboratory is it tells you a lot about the synthetic biologist and his or her prejudices, right? But when you go out and search for life, it also tells you a lot about the biologist, the prejudices of the person who's doing the searching, because that's the only thing that they will discover.
Yeah, but of course, look, we developed such a piece of software for astrobiology that is called Hypothesis Browser. And the way it works is that you put your arguments for something, for some hypothesis, like let's say there was RNA world, and you're going to be very prejudiced, and you will be supporting this, and then there's somebody else will put arguments against, and all taken together, it will be a balanced view. And yes, I mean, if you try to build a cell, an artificial cell, it will be biased by what you think. And what Kate thinks also that will be a bias. But if there is 20 laboratories that put their own biases into play, it's quite possible that you'll get a pretty broad and pretty kind of objective picture of what synthetic biology is capable of.
So
Yeah. Yes. Oh, okay.
So I have a recommendation. NASA has a new plan for Mars sample return. Now I don't know if it'll happen or not. I've been working on that subject for about 40 years, and it hasn't happened yet. But there is a chance, and the certainty is if it does happen, it'll be the one and only chance for decades. And I think it would behoove us before we do the only chance for experimentation. We need to take microorganisms in a carefully prepared package there and make them take the round trip to Mars and bring them back and study them. This will be our only chance to actually get some experimental data on the subject. It relates to transpermia, of course, but it also relates to the future of human missions on Mars. And I know there'll be a hue and cry from the community. I've worked on this. We actually did exactly that on the Russian Phobos sample return mission. Unfortunately, it never made it out of Earth orbit. But this is something that can be done. It can be done carefully, and it will be the one and only chance for decades. So that's my recommendation.
Did you talk to Lisa Pratt about it?
Did I talk to the what?
To Lisa Pratt.
Of course we talked to the press. As a planetary society, we did nothing.
No, to Lisa Pratt,
Planetary Protection Office.
Oh, Planetary Protection.
Planetary
Yes,
Protection.
we talked to Planetary Protection, and they had a lot of hand wringing, but at the end of the day, we did it, and they were fine with it.
So can we actually, maybe that's something that RP could comment on. Is it even possible right now?
Well, I think with the engineering to do it is there. There's probably not enough political will to do it if it's a government agency. I think it would take getting everyone involved on a pretty broad scale level with cost sharing. I think it comes down to doing that. I think scientifically we're there, and in experimental detail, I think we could pull it off. But funding it, getting it funded would be another story. I think that it would be, you would have to convince a large body of people to cost share and be able to do it in a practical sense. But I do. I think this community can pull it off. What do you guys think?
I think so.
Who can? Yep.
Yeah. Kind of along the lines of whether or not Mars sample return will ever happen, the strongest argument I think I've heard for doing Mars sample return at all is that this could be the last chance for us to get uncontaminated samples before humans land on Mars, provided SpaceX gets there anywhere near their timeline that they're predicting.
Okay.
Exactly.
Okay, so I just wanted to express some doubt. I mean, there is this general theme that was elaborated on of trying to create synthetic biology and use that to transfer information and so forth. And frankly, I don't really understand the obsession with biology because, you know, I look around and I see all these electronic devices, and I think we're on the brink of actually replacing biology, which is clearly not optimal. You know, there is a lot of junk in the DNA, and so we can do better, right? We can build things that operate more efficiently, and in order to transfer information, that, you know, we can build devices that will not be biological at all. And if you think about it, you know, a civilization that had 1 billion years would probably do the same, and therefore the biological phase that we are all obsessed with and we are talking about forever, perhaps just because we are biological, may be just a phase that that we have to overcome eventually and build something that is much more robust to environmental conditions and will be made of silicon or something else. And that is our future. It's not biology at all. So why obsess with biology? That's my question.
Okay, let me take a crack at this. You see, was this really electronic thing? It's artificial intelligence. And artificial intelligence, half of it is true. It's artificial. It's not intelligent. I mean, it's just you know, neural networks put on top of a very large database. Recently, I had lunch with a guy whose name is Michael Frumkin, who is the head of machine learning at Google, and he was telling me this story about they were analyzing images from Stanford, from medical school at Stanford, and that was just to distinguish based on those images. That was to distinguish people who have different types of diseases and so on. And they had a number of classifiers, and they could not. And the algorithm did a very good job at doing this. And was quite confident that clustered things according to a rule. They didn't know what the rule was, and it didn't correlate with any classifier any index that they had. Finally, they found out what it correlated with. It correlated with who took the picture. So it tells you that your fascination with this intelligence and with this ability of non-biological systems to do wonders, it will not happen in your lifetime, that they will do better than biological systems.
The technology evolves on a 2-year e-folding time, and you are a little impatient here because just wait a few thousand years, and you know the
yeah. If I wait 2,000 years, maybe
compare that to the evolution of biology.
Give me this 2,000 years.
Biology had billions of years. If you give that to technology, you know, you will never recognize what you're talking about.
Well,
If that's
but
a
look,
policy,
we
give it
talking
2000.
about we talking about what's going to happen in our lifetime. Maybe not. Let's say the lifetime of the youngest people in the room. Yes, I mean, what's going to happen in 2,000 years? We can speculate as much as we want to, because there will be no test who is right and who is wrong.
There will be. Just wait.
No, we will never know that. And but in our lifetime there will be tests, and the reason why we are interested in biology is that so far there is a number of tasks that biology can do better and will be able to do better in our lifetime.
But let me make a point here.
I think we had a cube.
Oh,
we still have time. They want to make a point about these things.
yeah, just briefly. We have broken free of Darwinian constraints and are entering a Lamarckian phase, right? Up until now, your slow biological evolution has been because the only way we can maintain our genetic information is by having kids die, and the only way we can improve it is by having even more kids die. And you're now at the position where you are able to do prospective changes and do prospective evolution. Now, that's partly because of your instrumentation technology and all the artificial intelligence and all the rest of it, the large databases and the neural networks. So you're going to see an acceleration in biology as well, because for the first time, we don't have to do that. We can actually do something better. And the only real question is whether the political body of Homo sapiens not evolved for that technology will use it successfully or not.
Maybe I'm changing the subject.
No, I think we had.
Oh,
Ben,
yeah.
Ben?
Go ahead. If there's
Go ahead.
Me?
Yeah.
Oh, I might be changing the subject, sorry, because I'm a little slow. I'm still cogitating some very impassioned, forceful speeches about immigration. And they didn't sit well with me, and I'm trying to figure out why. The panel up here was very focused on ethics and metered and careful, and then we had Charles get up and make his impassioned speech, and Zubrin behind me. In high school, I gave a speech sponsored by the American Daughters of the Frontier or something like that, I don't know. I was a pioneer girl, you know, the descendants of pioneers who came here to California for the gold rush. It was very much a part of me and who I am. And so this idea of new frontiers and how American democracy was created is very much a part of my upbringing. And that was long before I even had an interest in science. But this idea of space exploration, pushing, looking for new horizons, new frontiers, and how it affects us as a species is amazing. It's beautiful. It's fundamental to who we are, and I'm all for that. But I do not think that that's an excuse for us to just ignore our history and the horrible things that we've done. I think that we are mature enough to learn from our mistakes and to change the narrative and to go forward and explore for good reasons in a metered way that's careful and takes into consideration, you know, some kind of evolution towards goodness. And I would like to see that narrative be developed more and explore for the right reasons. That's all.
Can I talk now? Yes. So I wanted to respond first to the comment about SpaceX. There's no reason SpaceX should contaminate Mars any more than we would through a NASA agency. They're subject to quite a bit of their income from the federal government, and there's no reason we should abdicate that to them just because they have a banner that's called commercial, whereas there's other people involved in government agencies. So that's one comment. The other comment is a big change in subject, and I just wanted, because it's something I was thinking about before coming here, and a couple of other speakers have already alluded to it, and that is, why don't we look in our genomes for evidence that, in fact, an intelligent civilization did seed us? And it's something I don't believe is true, because I think they would have probably started out with oxygenic photosynthesis instead of making us wait 1 billion or so years to have it show up. But nonetheless, it's not that hard to do. And NASA Ames has supercomputers, and In fact, there's dozens of organizations now that do it. It probably doesn't even take that. And in particular, I was thinking pi would be a good one. We know that value of pi now to trillions of significant digits. So it's hidden somewhere in the genetic code. We have a good chance of finding it. There are problems because the code evolves, and so you wouldn't probably ever have a perfect match of all the digits or all the significant ones and zeros. But nonetheless, you could probably have a chance to search enough to say there is or isn't a match out to some significant number of digits that says this is either just random luck. You know, the numbers 314 are going to be in the code plenty of times. But you go out 10 or 15 or 20 digits, and it's going to be unlikely that it'll be there by random chance. And holy cow, if there is that number there, then you start looking for all sorts of other things that could be communication. It wouldn't cost a lot of money, and the ramifications would be enormous if it was really true.
Just to clarify from what I was saying about SpaceX, I'm not saying that they're going to go and irresponsibly contaminate Mars because they're not complying with planetary protection. I just mean once we have humans there, it will be very hard to not contaminate Mars in some way.
We're having meetings once a year on planetary protection issues for human settlement, and it's not clear that that's not the case, that we can't keep the same standards. Effective as vacuum. I don't think we should give up a priori on that.
I think just reinforcing both of these points, the call for how do we actually do this in a values-driven way? How do we respect the various interests that are sometimes conflicting and definitely exist, but still rise up and realize we need to reinforce these global institutions and norms so we don't say things are inevitably going to happen, but actually take an active role in shaping them. And they are not going to be, these are not easy problems, and figuring out how to manage global institutions in order to negotiate everything from the high-level goals right down to the minuscules of the protocols that are going to keep those interests safeguarded is something we're never going to solve. We have to continue to reinvest in, maybe even more frequently than a year timescale.
Lucianne?
Yeah, I would just like to pick up not only on this part of the conversation, but where Natalie left off and Bob's comment earlier. In thinking about these questions of the ethics that we bring to our exploration of the solar system and possibly even beyond, one of the most interesting jumping off points that I have had posed to me in the past year or so deals with this issue of time, right? Like we've been talking about how do we think on this truly cosmic time scale, 100 years, million years, whatever it happens to be. And this question that was posed to me was by Chanda Prescott-Weinstein, who said that it's interesting to think about what futures we might preclude through our actions. So this goes to, for example, since Bob brought up the colonization of the Americas, what futures did that preclude from happening? It precluded many things from happening, and I think it is not correct to say that inventions of the modern world might not have happened other ways. So I think that speaks to kind of the inevitability that is often folded into a lot of these questions. And I think these are very difficult, but when we're talking about, a future where we might be faced with the question you raised, Tanya, of, how do we decide at what point humans go? Is it fictitious to think that there's a moment that we will know that there's no life on Mars? And to your point, Megan, about moving beyond this idea of containment and instead evaluating all of these systems in the deeply enmeshed way that they actually exist in the world, I think it would benefit us to think about some of these not only scientific questions from a long-term timescale, but also some of the social and ethical questions as well, because we do have this impact that we might make very long down the road in ways that we don't fully understand unless we're prepared to deal with that.
Okay. Anyone wants to do a last comment?
Just one last comment, okay?
Well, negative evidence is always very difficult to give. So you raise this issue, will we ever know that there is no life on Mars? And probably you can give a negative evidence in the context of mathematical theorems. In some cases, in laws of physics, which are very, very strict and universal. But in most cases in natural world, I'm afraid we'll never know that. And you know, when you raise this issue of what kind of future is precluded and so on, that's a very deep issue that philosophers and historians were worried about, that there is this one idea, that those two schools of thinking is that one idea is that one person can change the course of history. And there were people who were changing the course of history for better or worse, yes, like Hitler or Napoleon or so on. And then there is this other way of thinking that says, no, there are some historical forces that cause things to happen. And if this person doesn't do it, then somebody else does it. And there is, so it doesn't really matter. There are those very fundamental social forces that push the world in this direction and not in some other direction. And if you take this approach, there isn't really very much we can do, yes?
I just wanted to emphasize, I really like that articulation of the ways in which to think about this, and also that we're not in a static world where we're just making one decision and not falling into this trap of assuming that inevitability of certain types of actions that cause us to just move faster. But I think this also argues for the fact that we need to be, you know, enmeshed in historical other types of conversations and that really situate our current imaginations about what we should be doing and also limit those imaginations often. I think it's, again, an opportunity.
So let's have our last cube comment.
Okay. I think there's a false dichotomy that's been presented here. You know, to go to Mars or not go to Mars, to the fella who works on, is interested in Europa, to go to Europa or not. People are going to build habitats over the next couple of centuries. And very soon, just like all those tiny little islands in the Galapagos, they're going to radiate quite rapidly because that's what evolution does. And every one of those habitats, as they get bigger, you know, we're going to learn how to make them self-sustaining. And each one's going to become a little world. And that's without worrying about stepping on a planet and contaminating that planet at all. We'll be in space. So you're talking about the emigration of life from Earth. And, you're going to have all these separate ecologies that are going to diverge as rapidly from one another. And they'll all be free-floating inside asteroids and whatever, out to the Oort cloud, without contaminating Mars or Europa. So think about that. That's a precursor to real interstellar travel. Thanks.
That's a real emigration. That's kind of like those ships. So maybe we should start wrapping up. Maybe we should start summarizing if anyone would like to do it in 30 seconds.
Oh boy. I guess following up on the last comment, people ask me, are we going to Mars? Are we going to Europa? Are we going beyond? Of course we're going to do all these things eventually, we as humans. But I think the interesting question, and leveraging off what Frank Dragoy says about finding intelligent life, the question really is it going to happen in our lifetime? I have absolutely no doubt all these things will happen in humans' lifetime. It's just, obviously we want it to happen in ours because otherwise it's sort of irrelevant for us.
Yeah.
Well, I think that we do need to undertake this effort of exploring our, space with our life. But I agree with all these concerns that were raised here, that we have to do it thoughtfully, not to cause more damage than good.
I just valued this conversation. I think that what I'm trying to take out of this really is I think that how we get there matters, and the investment in these processes and realizing that they are slow and take a long time, and there's value in that. And there are a lot of different opinions, values, imaginations that are not in this room, or maybe that we haven't even explored yet. And I think that exploration is useful as well.
Yeah, I think a final thought for me, there's sort of a question about scale and our concept of time. I think about a lot of this. There's a book written a few years ago called The Human Scale, and I think one aspect that I took away, it was that there's a quote in there that says, man is a measure of all things. Like there's a certain amount that we can really see in our day. Like how far can I walk in one day? Can I see the edge of a town? That's about how far you can walk in a day. You know, so what parts of, you know, city life, I'm kind of a country boy if you guys can't tell from my accent, but like if you go to a city and you say, well, like how tall is the tallest building? You know, you think about that in terms of trees. What's the size of a tree? You know, above a couple of trees, it starts to feel a little bit uncomfortable. So I was thinking about if we emigrate, you know, what is this concept, on what scales are we willing to accept in immigration of life? You know, do we want to do this immediately? You know, is that seen as natural or unnatural? You know, if we want to do this over a succession of a few thousand years, is that going to be seen as natural or unnatural? And who's to decide? I think it's helpful that whether we decide if this is a natural progression of our own evolution, it's through conversations like these. And I think I value the diversity of thought up here, and I think that's been a really great conference so far. So thank you guys.
I think we're at a really unique point in terms of our technology and also our social awareness to think about how we're going to do this and do it in a better way than we've done it before. And so we can have these conversations now that weren't had previously and try to be better about how we expand beyond Earth.
Okay, thank you very much. Thanks all of you guys, and thanks to all the cube holders. And I think we are done with that session. And Penny.